we are going to discuss another interesting topic of cell cycle, that is cell cycle arrest. In the previous video, we have already discussed what is cell cycle, what is interphase, what is mitosis, and what is meiosis 1 and 2. We have already discussed in detail. So if you have not watched that part of the video, I do recommend you to please to watch the first part of the cell cycle where I have discussed about interphase. Second, I have discussed about mitosis. Third, we have discussed about meiosis 1. And fourth, we have discussed about meiosis 2. So before coming to this part of the video, that is cell cycle arrest, you need to have a good understanding about this four part. Why? Because cell cycle, during the process of cell cycle only, the cell cycle arrest. Arrest means what? Stops. So why do they stop? That we will understand now. Okay. So let's begin. If you have understood, in the previous videos we have discussed, in the first part we already discussed that the cell cycle occur in a cyclic manner. That is, first the G0 phase will come, then G1 phase, then S phase, then G2 phase, and then the M phase. Again this occurs in a cyclic manner. This cycle goes continuously unless and until there is any error during this process. If there is any error during the cell cycle, then the cell cycle will stop, arrest will stop and repair it and then move forward. For that purpose only, there are certain checkpoints are present during each stages. Okay, during each stage of the cell cycle, there will be certain checkpoints will be present. What is checkpoint? You can simply understand in the simple meaning what is checkpoints. Checkpoints is nothing but is like a gatekeeper. What is the role of the gatekeeper? Or to check if everything is going on smoothly or not. So the checkpoints do the same. So second point, third is, if you remember, as I have already told you, G0 to G1, G1 to S, S to G2, G2 to M, and then again. This is the cell cycle. So this G1 checkpoint, where it will be present? This G1 checkpoint will be present here before the cell enters the S phase. So the G1 checkpoint will be present at the G1 to S transition stage, and during this phase the checkpoint will be present. Next, where will be the G2 checkpoint will be present? Before the cells enters the M phase, that is the mitotic phase, the checkpoint will be present, that is G2 to M phase. Next, spindle checkpoint. You understand what is spindle fiber? We have already discussed when we were discussing about the mitosis. So spindle checkpoints, during the metaphase spindles were formed. After the metaphase, what was happening? Anaphase was formed. So from the metaphase, the transition between the metaphase to the anaphase, these checkpoints will be present. Why? If you remember, I have already discussed that during the metaphase, the spindle fiber will be attached to the chromosome. Why? Because during the anaphase, they will be pulled apart from each other to the opposite pole. So that at the end of the anaphase, during the telophase, they will be divided into two daughter cells, which we have already discussed in the mitosis as well as in the meiosis. So this spindle checkpoint will check whether the spindle is attached properly during the metaphase or not. So the G1 checkpoint is present in the G1S transition, G2 is present during G2 and M transition. Transition means before entering the S phase, this G1 checkpoint will check whether everything is okay or not. Similarly, G2 checkpoint, before entering the M phase, this G2 checkpoint will be present to check everything is okay or not. Similarly, spindle checkpoint, the same thing happens. Okay, so this is the highlight, or this is the three types of checkpoints. Now we are going to discuss each one of them in detail. The first checkpoint, that is the G1 checkpoint. Okay, if you remember that we have studied, the G1 is known as the first growth phase, during which what what happens? The cell size grows so that it will enter into the S phase, and if you remember, during the S phase the DNA synthesis occurs. So you can also remember like this: whatever the things are required for the DNA synthesis during the S phase will be prepared during the G1 phase. Okay, as I have already told you, if you are cooking something, first of all you will collect all the ingredients, then you will cook. Similarly, before entering into the synthesis of the DNA phase, whatever things are required, whatever the cells need to undergo will be done during the G1 phase, which we have already discussed. So what does this checkpoint do? It will check whether, first it will check the cell size, whether the size of the cells is enough big or not so that the DNA synthesis occurs. Second, it will check about the nutrients, whether the enough nutrients are present or not for the DNA synthesis, for the process of DNA. Molecular signals. What are molecular signals? That is the internal signals, that is the growth factors, like the growth factors are properly coming into the cell or not to ensure the cell size is growing. Okay, all these factors will be done, all these things will be done by this G1 checkpoint. Whether the cell size is large or not, whether nutrients are present or not, and if the cellular molecular signals, that is the growth factors, are coming or not, all these things will be checked by this. DNA is broken or not. Why this step is essential? Because after this G1 phase, it will enter into the S phase, and if the DNA will be broken and synthesis occurs, then we will get a mutant type of DNA or broken DNA, which will be very difficult for the further process. So, to ensure that the DNA is proper or not, it will check whether the DNA is broken or not. Also, these 4 steps, after checking all these steps, then it will allow the cell to enter from G1 to S phase. If any of the following deformities is present in the cell, then it itself will stop here, and it will take some time to repair all these things, then it can move further to the S phase. Okay, so this happens what in the G1 phase. I have already told you this is from the transition from G2 to M phase. That means M means what? Mitosis phase. Okay, we have discussed already. So before undergoing mitosis, all the steps which are required, that will be checked during this G2 replication have occurred or not properly. DNA replication have occurred or not. Why? Because before this G2 phase was the S phase, that is DNA synthesis was occurred, that is DNA replication occurred during S phase. Whether DNA replication have occurred properly or not, so that when during M phase, when they will divide into 2 daughter cells, then each daughter will create equal amount of DNA. It will check whether DNA replication is DNA attached to the protein or not. Okay, it will check whether the DNA is properly attached or not, and also it will check the cell size. Again, it will check the cell size, and after all these things, if checked, if everything is okay, then it will move to the M phase. Otherwise, the cell will stop here and will undergo the process of apoptosis. What is apoptosis? We are going to deal with this topic very soon. It will undergo, that is programmed cell death. The cell will die and will again the cycle will start. Or else what will do? It will repair. Either it will go on apoptosis process or it will repair if there is any deformities. If any DNA repairing process will occur, certain proteins will be activated to prepare this proteins, DNA replication, then it can smoothly enter into the M phase. So remember, this checkpoints is only to ensure whether everything is going on smoothly or not. Because this cell cycle is very, very important. Okay, by the end of the chapter, meiosis only, I have told you what is the significance of meiosis. And also mitosis, you know what is the significance. If you know the significance, then you can clearly understand why these things are required. Because if you don't check everything is going on smoothly or not, then any type of error can completely destroy the structure. Okay, can completely harm the cells. Okay, that between the transition stage of metaphase to anaphase. As we have already discussed that during the metaphase the spindles will be attached to the chromosomes, and if you remember the structure, as I have already told you, this is the chromosome structure. They will be arranged in the equatorial plane during the metaphase stage, and to here this is the centriole, and to here the spindle fibers will be attached. Spindle fiber will be attached. Why? Because after this stage, anaphase, they will be pulled apart and they will be separated out to the opposite pole. And after this anaphase, telophase, they will form the two daughter cells. So to check whether the spindles are attached to the chromosomes or not properly, this spindle checkpoint is present. It will also check if any chromosome is moving freely or it is attached or not. That also it will check. If there is any deformity, then it will correct it, and then it can move to the anaphase. Okay, once it enters the anaphase, then the spindle fiber will contract and it will pull apart, and then division to check or to repair this piece. Okay, this is the introductory part, and this we will continue in the next video also. So cell cycle regulation, two things are required. One is CDK, another one is cycling. What is CDK and what is cycling? The cell cycle to occur smoothly, two proteins are required, or you can say one protein and one enzyme are required, which controls this cell cycle progression. We have already discussed what is cell cycle, and from one phase to another phase how it is moving because of these regulatory unit. That is, one is known as the cyclin, which is a regulatory protein, and second one is CDK. The full form of CDK is cyclin dependent kinase. So from the word kinase itself you can know that this is an enzyme, and dependent means it will depend upon the cyclin for this activity. So both the cyclin as well as CDK will act in the regulation of cell cycle. For the progression of cell cycle from one stage to another, these two will be required. Then we will first discuss protein. Why it is required? For the progression of the cell cycle it is required. Okay, R-pinothe was discussed of its different concentration during the cell cycle. So during the cell cycle, the concentration of cyclin varies. That's why it is called cyclin. Okay, and if you know how many types of cyclins are present, there are around 20 different types of cyclins are found in the cell cycle progression, and many of them functions are still unknown, but these are some of the points of the dependent kinase, and it belongs to the kinase family, means it will undergo the process of phosphorylation. What is phosphorylation? That is transfer of phosphate group to activate certain cell cycle, then phosphorylation is required. So who will do this cell cycle dependent kinase? It will depend upon the cyclin for its activity, and it is an enzyme which belongs to the involved in the transcription as well as in the mRNA process. So that's all for today. In the next video we are going to discuss about the cyclin and CDK in detail, some other points. Cell cycle regulation we will be discussing in the second part of the video, and hope this part of the video was helpful to you. If you have any doubt regarding this video, do ask in the comment section. If you are new to my channel, do like, share and subscribe my channel, and I have made many. That's all for today. Have a nice day. Thank you.